Hello, everyone, and welcome to another exciting episode of the Biologic Podcast. Today's show is episode 112, and today we're going to talk about spiders, the true spiders of the order Araneae. It's been a long time coming, too. If you've been listening to this entire series on arachnids sequentially, then you might feel a bit of suspense right about now. In every episode so far in this series, from early arachnid evolution to the many mysteriously divergent basal clades that exist today, I've just been merely mentioning the true spiders, dropping small bits and pieces of information, a physiological comparison over here, a phylogenetic relationship over there, and so on. Well, now it's finally time. It is time to dig into the clade of true spiders and explore the breathtaking biodiversity and incredible lifestyles of these small but critically important creatures. Of all the arachnids, the spiders of the order Araneae are the most diverse, with almost 50,000 known species that have been discovered and described by taxonomists, and there's tens, perhaps even hundreds of thousands of species that are estimated to exist but are undiscovered or undescribed. Because of this really impressive biodiversity and the extraordinary ecological value of the spiders, I'll be spending not one, but 2 episodes covering them. That's right, this is going to be a 2-part miniseries. In this episode, in part 1, we're going to explore the anatomy, physiology, behavior, and ecology of the spiders, and then we'll explore 2 of the oldest, most basal clades, the Mesothelae and the Mygalomorphae. In part 2 of this miniseries on the spiders, or episode 113, we'll look at the more recently derived clade Araneomorphae, which includes the vast majority of all living spiders. That should be a super interesting episode too, and of course we'll round out the arachnid series with a final episode on the relationship between humans and arachnids. Now that final episode will be largely focused on the spiders, as spiders are, by far and away, the most iconic arachnids. They're less destructive and harmful, and therefore less hated than the ticks and the mites. They're more geographically spread out, and thus more familiar to more human cultures than the scorpions, and they're far more diverse and appealing than their Europyge and Amblypyge cousins. One last thing. If you want to get a bit more background information on the arachnids before we get started, I recommend going back and listening to episode 87. Let me briefly summarize some of the main points that are relevant for today's episode. 550 million years ago, the protostomes were undergoing significant evolutionary changes. The Spiralia clade emerged and began to diversify into primitive annelids and mollusks. Ancestral clades of primordial ecdysozoa evolved a variety of new features, like exoskeletons, jointed limbs, body segmentation, and the ability to molt, to shed an old exoskeleton and grow a new, larger one. With these freshly evolved traits, the ancestral proto-arthropods could endure harsher climates and more rugged habitats. They could move deeper inland, away from the oceans, to colonize drier, hotter areas. One of the new groups that emerged around this time, around 500 to 510 million years ago, were the chelicerates, arthropods with 2 large clawed mandibles mounted in the front of the head, right above the mouth. In some clades, the chelicera are small and used merely for handling food immediately before ingestion, before it gets put into the mouth. But other clades, like the ancient Eurypterids, had large chelicera that have been adapted into fully fledged arms, with big pincer claws on the end. Other early chelicerates include the horseshoe crabs, the mid-Silurian Ophicolis, and ancient Pycnogonida sea spiders, like Palaeoisopus and Heliostes. Approximately 468 million years ago, a chelicerate clade called the Planeturga emerged. These animals were similar in morphology and ecology to horseshoe crabs, although the highly segmented rear section of the body, the Opisthothelae, appears to be moving away from the head section. For example, the first section of the Opisthothelae, closest to the head, had evolved to lose its pair of appendages, creating a distinct gap, like a neck or something. After some 30 million years of evolution, the first true arachnids emerged from within this Planetarga clade, about 435 million years ago. Some of the earliest arachnid groups include the Opilionese harvestman spiders, the Rhysnulae pseudospiders, the Solifugae camel spiders, and the Acariforms and the Parasitiforms, more commonly known as the mites and the ticks. There was a more divergent clade, which produced the scorpions and the pseudoscorpions, and then of course the Tetrapulmonata. This last group, the Tetrapulmonata, is defined by the presence of paired book lungs in the Opisthothelae, which are used for respiration. They're like centralized respiratory structures, as opposed to the more decentralized tracheal network that you see in insects, for example. Within this Tetrapulmonata lineage, the more recently emerged Schizotarsata lineage would produce the Europyge whip scorpions, which appeared in the fossil record about 360 million years ago, and the Amblypyge whip spiders, which appeared about 315 million years ago. There's another Tetrapulmonata lineage, even older than the Schizotarsata, which is called the Cercodiastida, which emerged about 385 to 390 million years ago, in the early Middle Devonian period. The name Cercodiastida is Greek for silk worker, which refers to the emergence of silk-producing organs in this group. Some of these Cercodiastida clades that existed in the Devonian, such as the Araneae, have a remarkable spider-like form, but they also retain some more primitive features, like a long, thin tail that we also see retained in the closely related Europyge and Amblypyge. The oldest known fossil is the Attercopus fimbriungueus, which is 386 million years old and exhibits spinnerets on the underside of its abdomen. So here we can see the combination of older, more primitive traits shared with cousin lineages, like the long, thin tail, with newer traits seen only in this specific silk-worker clade, like the spinnerets. Things would continue to change as time went on. Roughly 320 million years ago, a new Cercodiastida group emerged, the Araneae. These are the first true spiders, and as we'll soon see, they've come a long way from the horseshoe crab body forms of their ancient ancestors. The modern spiders of the Araneae have a body with two major tagmata, or body segments. There's a cephalothorax, or a prosoma, which is essentially the head and the chest, so to speak, and then there's the opisthothelae, which is the hind segment, more colloquially referred to as the abdomen. So let's explore spider anatomy a little bit, beginning with the cephalothorax. This body segment includes the head, the eyes, and the brain, as well as the chelicera and pedipalps around the mouth, and inside it contains the main stomach cavity. Perhaps the most characteristic feature of the spiders are their 8 eyes, arranged in 4 pairs near the top front region of the cephalothorax. The 2 primary eyes, the big ones right in the middle, are capable of forming images. These eyes also have retinas, sometimes with multiple layers of retinal tissue, and muscles that can move them around slightly, allowing the eyes to produce highly detailed 3D representations of the world. Some spiders, like the jumping spiders, have extremely high visual acuity, greater by an order of magnitude than any predatory insect. All of the other eyes, the so-called secondary eyes that are arranged around the head and face, are thought to be derived from ancestral compound eyes still retained by insects and some other arthropods. These secondary compound-derived eyes are less complex. They don't have muscles, and they can't form images. They're mostly used for detecting the direction of light and shadows, and for picking up gross movement in the periphery of the visual range of the larger primary eyes. The spiders are also uniquely characterized by the structure of their brain. Among most arthropods, the nervous system includes 2 nerve cords running the length of the body, studded with ganglia in each body segment. Ganglia in the front of the head fuse to form a ring around the esophagus, and this ring is like a primitive brain. This is the case for most arthropods, but the spiders have evolved even greater centralization of the nervous system, With the exception of the oldest Mesothelae lineage, the spiders generally fuse all of their ganglia into a single brain organ in the cephalothorax, with no ganglia at all left in the abdomen. This sophisticated, centralized brain structure allows the spider to exhibit more complex behaviors. Spiders in general are relatively intelligent animals, especially compared to other arthropods, like, say, millipedes or certain insects. Depending on the species, the spider can mimic the sounds of the wind or a potential mate to lure a prey animal out of hiding. They can also engage in complex mapping and route planning to get to a goal. Like, they can see something that they want somewhere in the distance, they can map out a path through the environment to get there, and they can follow that path to the goal, even if, during the course of following the path, it takes them out of visual range of the goal. They can still remember their goal, their task, where it is, and how to get there, even if they can't see it. And that's actually a pretty sophisticated behavior that a lot of larger animals with larger brains can't do. Furthermore, they can also learn through risk assessment. They can look at a situation, see how their goals fit into the situation or the environment around them, and make judgment calls about what pathway or what activity or, what particular behavior might have certain risks associated with it, and which risks are unacceptable, and which risks perhaps are acceptable. So if they're in a situation where any course of action has risks of various severity, they can identify those risks, calculate the relative riskiness of them, and then choose the course of action that has the least risk to, say, their health and fitness or their life. Spiders can learn through trial and error. They have working memory to remember mistakes they made, and they have the cognitive flexibility to think of other approaches to a problem, other ways to strategize to get to a goal, while integrating information from that previous failed attempt. And again, a lot of this stuff sounds really simple and basic, but it actually represents very sophisticated cognitive tasks, stuff that is really surprising to see in a creature like a spider. And if that wasn't enough, some spiders even have a conception of numbers on par with a 1-year-old human child. Jumping spiders in the genus Portia are believed to be the smartest of all the spiders, a trait that's suspected of being related to, or even caused by, their extremely good vision. Now, most spiders have relatively small pedipalps that do little more than handle food. The Chelicera do much of the heavy lifting. They're the primary appendages used to initially capture prey, as they're equipped with venom glands and sharp syringe-like fangs. The vast majority of spiders are venomous, but some, like the Uloberidae and the Liphistiidae, have evolved to lose their venom, and they hunt using their silk, or through raw brute force. To my surprise, spiders are fluid feeders. They can't ingest solid food particles, so they use filters, such as a mustache-like structure above the Chelicera, to block the particles from entering the mouth. Some species of spider, much like the velvet worms, will inject digestive enzymes directly into their prey to liquefy them, so that the soft, fluidy innards can then be sucked out. In yet other species, the Chelicera and the pedipalps create a small pocket where food is held and can be ground up and smashed into a juicy pulp. Then, much like drinking the juice out of an orange you've pulverized in your hands, the spider slurps up the wet remains. When food is ingested, it's moved down to the stomach, which is still in the cephalothorax. The stomach continues the chemical breakdown of the food before pumping it into the digestive organs deeper in the cephalothorax and in the abdomen. This lower digestive tract contains many pockets or cavities called ceca, where the fluid can be stored and absorbed. Metabolic waste is converted into uric acid, which is secreted from the blood into the cloaca, where it's then excreted as dry pellets. Curiously, the spiders don't have antennae. Where almost all the other arthropods do have antennae, even the crustaceans, the spiders don't. This is because the first head segment that would form the antenna actually disappears in spiders during embryonic development. Instead of antennae, spiders typically use the pedipalps and Chelicera around their mouths to sense the world in front of them. They also use their setae, bristly hairs that provide tactile feedback to gain more sensory information about their environment. The underside of the prosoma is a pelvis-like platform from which the legs extend, 2 pairs pointing towards the rear and 2 pairs pointing forwards. The prosoma has a single curved carapace plate on top, and 2 flat plates on the bottom, between the joints connecting the body to the base of the legs. Each leg itself has 7 segments, including 2 segments analogous to a shoulder, and 1 segment each analogous to the humerus, the elbow, the forearm, the hand, and the fingers. That last segment of the leg, analogous to toes or fingers, are instead multi-pointed claws on the spider. Now these are all the parts found on the cephalothorax. The other body segment, the hind segment called the opisthothoma, or the abdomen, is generally larger. It's more globular in shape. It's unsegmented, and it contains the heart, the hemocoel, and much of the vascular system, including an aorta that runs up the cephalothorax to vascularize the stomach and nourish the brain and the legs. The opisthothoma also includes the rest of the intestinal system right down to the anus, as well as the gonads and reproductive organs, the book lungs, and the silk glands. Speaking of the gonads and reproductive organs, spider reproduction is a very interesting process, with many unique behaviors. Lots of arthropods are known to create spermatophores, which are gooey balls of sperm and protein that can be ingested by the female or inserted into her cloaca in order to reproduce. Spiders don't really do this. They don't really use a spermatophore like that. Instead, the male will create a small web, a small little bit of silk to be used like a wrapper or a napkin or a container or something for holding ejaculated sperm. The male spider will load the silk-wrapped sperm into an organ called a palpal bulb, which is located on the end of his pedipalps. Thus prepared, the male sets out to look for a mate. He'll generally use sights and smells to find a reciprocating female. For example, he may smell the webbing of a female, and chemical cues in the webbing can give information as to the female's species and reproductive readiness and general health. If a receptive female of the same species is located, a complex mating ritual begins. Now this varies a bit between species, but in general, the purpose of the mating ritual is to subdue the female. She's generally larger and more dangerous than the male. The male is at risk of being eaten. Sometimes, if there's a male out looking for a mate, he'll specifically look for a female that has just recently molted out of her exoskeleton, because she'll be tired and will be less likely to attack him, or if she does, she'll be less likely to catch and eat him. Anyway, the complex mating ritual, which typically includes specific patterns of touches on the female's body and or on her web, will soothe and hypnotize her, allowing the male to get close enough to climb on top of her and manually deposit the sperm in his palpal bulbs into her genital pore on the underside of the abdomen. Within the female reproductive organ, there are chambers called spermatheca, which store the sperm. So fertilization doesn't actually happen during or immediately after the act itself. The stored sperm is released at certain intervals that sync up with the ovary releasing eggs and sending them down the reproductive tract. And this can occur long after, like days or weeks after the actual mating event. There is one exception to this, the Herpactia sadistica, a spider found only in Israel, which doesn't gently deposit sperm into the genital pore with its pedipalps, but instead it uses an aggressive syringe-like traumatic insemination method, where the sperm is injected directly into the ovaries, leaving rows of holes punched in the female's abdomen. This traumatic insemination strategy is believed to have evolved as a response to the spermatheca. The sperm storage chambers generally let the egg get fertilized by sperm from the last male to inseminate the female. In the Herpactia sadistica, the males seek to avoid that gamble by directly fertilizing the eggs himself. After mating, the female often eats, or will try to eat, the male. Some males are able to survive multiple matings, But most males don't even mate once, and instead they just end up as food for a breeding female. Once fertilized, the females will produce a silk-lined egg sac that contains hundreds, if not thousands, of small eggs. Depending on the species, some females will hide the egg sac, like in a burrow or in the safety of a web, or they'll attach the egg sac to their legs or abdomen and drag it along with them. Or they'll just die shortly after mating and leave the eggs to survive and hatch on their own. In contrast, in other species, like the wolf spider, the female will keep the egg sac close by until the eggs hatch, and then will even parent the newborns for a short while, carrying them around and feeding them. This is something that the wolf spider has in common with many scorpions. And now, the last major part of the Opisthothelae and of spider anatomy that I want to discuss with you are the silk glands, the spinnerets, and the silk itself, which is incredibly interesting stuff. So the spinnerets appear as small, stubby, almost button-like appendages near the tip of the spider's abdomen. Each spinneret has multiple spigots, each connected to a silk gland, and there's 6 different types of silk gland that produces different types of silk. Think of it kind of like a fountain drink soda machine. There's lots of different faucets, each one connected to its own reservoir tank containing a specific type of soda. The metaphor doesn't end there. Soda is a liquid, and spider silk held inside the silk gland is also a liquid. Well, technically I guess it's more like a gel. Wait, what? You might be asking. How does a spider turn a liquidy, gooey gel into a silk fiber that has a ton of amazing physical properties, like an incredible level of tensile strength, despite its relatively low weight and density? How on earth does the spider turn the goo into the web? It's a really amazing process. Check this out. Within each gland, the liquid silk is basically a soup of proteins, including spidroin 1 and 2, which provide structural support to the final silk fiber. This gel-like soup of proteins is stored in a sac or a gland until the spider needs to spin a web or make a thread for some purpose or something. When this happens, when this need arises, the sac will condense, pushing the silk gel down a narrow tube called a tapering duct. The internal topography of this duct, including its narrowing corridors and tight turns, as well as a high-pressure, ion-rich environment, serves to align the proteins lengthwise, parallel to the duct. The high concentration of ions floating around here are used to remove water from the duct and increase the internal pH, making the duct environment more basic. This high-pressure, ion-rich environment, in combination with the mechanical forces of the duct, induce a phase transition where the gel-like proteins are squeezed and strained into solid fibers. At the end of the tapering duct, there's a spigot which has a firm hold on the fiber. The spigot can compress or expand to alter the fiber's diameter, and it also forms a seal that keeps in water so that the silk gland doesn't dry out or lose pressure. The fiber of spider silk itself is composed of alternating solid crystalline regions and looser connective regions that form an amorphous matrix. Other non-protein chemicals are integrated into the spider silk, each serving some kind of function. For example, there's a compound called pyrrolidine, which tends to absorb moisture from the air to keep silk fibers from drying out and weakening. And it also has anti-predator effects in that it discourages ants from coming and chewing on the web. Similarly, potassium hydrogen phosphate reduces the pH of the silk, making it a little bit more acidic, and makes it unpalatable for various fungi and bacteria. And then there's also potassium nitrate, which protects the silk proteins from denaturing in the lower pH, or the higher acidity environment. At the end of the process, the spider has produced some kind of silk, which usually has remarkable mechanical properties. These spider silk fibers have a very high ductility, meaning they can be stretched out to almost 5 times their normal length. But they also have a very high tensile strength, on par with high-grade steel alloys, but at about 1/6 of the density. They can retain these properties even at very high or very low ambient temperatures. Now, a few minutes ago, I mentioned that there's multiple types of silk, used for different purposes. This isn't an exhaustive list, but here's some of the most common types. There's minor ampullate silk, which is just used as scaffolding during the construction of a larger web. There's thicker dragline silk, which is used to construct the rim and the main spokes of the web. And a really awesome fact that I read is that dragline silk can contract when it gets wet, which is thought to be an evolutionary adaptation to the morning dew wetting spider webs, allowing them to tighten up and flatten out at the start of the day. Flagelliform silk is used to make the inner spiral that catches prey insects trying to fly through it. When a prey insect lands on a web, or more accurately, flies into the web and gets caught up in the flagelliform silk, the spider will approach it and wrap it up in aciniform silk, which is the toughest type of silk. It's meant to prevent the prey insect from chewing, slicing, or cutting its way out and escaping. Tubilliform silk is a special type that's used to wrap up eggs and create egg sacs. It's not the toughest material, it can be destroyed and chewed through, but it is the least flexible, and thus it offers some mechanical protection for the eggs, like a kind of hard case safety pouch that the eggs are kept in. Lastly, I wanted to mention that not all webs have the classic, flat, radially spoked web pattern. Some species produce orb webs, where the web is formed into a spherical shape, with a little pouch on top that the spider lives in. Orb webs aren't as effective at catching prey, but they might offer some kind of advantage with regard to concealment from predators, or resisting damage from the environment, like rain, or snow, or falling rocks, or scratching twigs being blown around in the wind. Some orb webs have various modifications, like built-in sensor fibers, or attachment lines running to the water, or even little silken ladders that help the spider crawl up and down from the ground to its orb nest. Okay, so that was a very brief, very superficial rundown of the general aspects of spider anatomy, from their head and their eating habits and digestive tract to their reproductive organs, their silk glands, and their webs. Now that we've established the basics of the spider body form, I want to return to their evolutionary history so we can explore some of the earliest, most basal Araneae lineages and set the stage for the more derived lineages of Araneomorphae in the next episode. Early in the evolution of the Araneae, perhaps around 310 to 330 million years ago, there was a primordial divergence that produced what has become the oldest surviving lineage of spiders in existence, a sister lineage to all the other spiders. This oldest lineage, this most basal cousin clade of the spiders, is the Mesothelae, or the Mesothelae. As one of the oldest lineages, its membership is heavily marked by extinctions. Most Mesothelae spiders have gone extinct over the course of their 300+ million year history, to the point that only a single family, the Opisthothelae, contains any living species. The Opisthothelae family contains 8 genera and approximately 100 species, which can be found across the jungles and river valleys of Southeast Asia, from Japan to Vietnam. The Opisthothelae are characterized by a number of primitive traits, both physiological and behavioral. For example, their many eyes are clustered together on a single node or bump in the front center of their head. Their 4 pairs of spinnerets are not fully internalized, but instead appear as stumpy appendages in the middle of the abdomen. The abdomen itself retains evidence of segmentation, similar to the abdominal structures seen in scorpions, amblypygi, and europygi. They have protective tergites, or a kind of armor plating running along the back of their abdomen, which is a trait that we saw in many solifugi sun scorpions, Riceinulae pseudospiders, and many of the true scorpions. Also similar to many scorpions, pretty much all of the Mesothelae spiders are nocturnal, They're nocturnal hunters that live in dark enclosed recesses or crevices, like those you'd find in caves, or thick leaf litter. Most species are so-called tube dwellers, referring to their tendency to construct small burrows or tunnels in the soil and leaf litter. These burrows have a hatch or a trapdoor at the end, typically made of woven spider silk and decorated with lichen and detritus, which conceals the burrow and the spider hidden inside. Within this concealed dwelling, the Mesothelae spiders will wait. They'll wait for some unaware prey animal to come stumbling by, their footsteps sending vibrations through the substrate, which the spiders pick up via vibration-sensitive hairs on their legs. Vibrations can also be detected via the spider silk hatch door, telling of a nearby creature. Many of these species, like those in the Liphistianae subfamily, also construct signal lines radiating out in all directions from the entrance of their burrow. These signal lines are typically single threads of silk laid out like tripwires. A prey animal wandering by will walk over these threads, creating a vibration that can be felt by the spider hiding back in its burrow. The spider will crawl up and poke its head out of the trapdoor, peering around to try and spot the intruder, and of course it'll know which signal line the vibration came from, so that kind of gives it a good idea right out of the gate. And then it'll dart out to capture the prey animal by surprise, with a downward strike of the chelicera. To the curiosity and frustration of field zoologists, the Mesothelae spiders are generally reclusive homebodies. The males spend much of their time in burrows, and generally they only leave to look around for a burrow with a receptive female inside. The females, on the other hand, are even more extreme. Once they find or build a suitable burrow in the shadows of the mossy undergrowth, they'll stay there, more or less, for the rest of their life, only darting out for mere moments at a time to snatch an unfortunate prey animal that wandered too close, inject it with venom loaded into its chelicera, and then drag it back into the darkness of the burrow, where it'll eat it. But aside from those brief interludes where the spider engages in ambush hunting attacks, aside from that, the female will spend her entire life hiding in the shadows of this burrow. Okay, so that was a very brief glimpse at the Mesothelae, the most primitive branch of the order Araneae. We see that it retains many primitive characteristics, like segmented abdomens, tergites, limb-like semi-external spinnerets in the middle underside of the abdomen, and nocturnal behavior, among other things. These traits are all considered primitive because they're similar to traits seen in older spider relatives like the Europygae and the scorpions, and because all the other spiders, all the other more derived Araneae lineages, don't have any of these traits. All the other derived spider lineages, which includes the vast majority of the almost 50,000 known spider species, fall into an unranked suborder called the Opisthothelae. Several traits define this group and separate them from the Mesothelae. For starters, the common ancestor of all Opisthothelae had evolved to lose the external segmentation on the abdomen. In these spiders, the abdomen has taken on a smooth, cohesive, singular shape, like a smooth fruit, like an apple or a pear, devoid of the creases and folds of segmentation seen in the more basal arachnids. Second, in addition to losing the abdominal segmentation, the Opisthothelae lost the tergites, the armor plating on each abdominal segment that we saw in the Europygae and the Mesothelae, among others. Third, the spinnerets have become fully internalized, so they don't hang out of the belly like stumpy vestigial limbs. And on that note, the Opisthothelae have evolved a more efficient location for their spinnerets. Instead of their belly, or the middle of the underside of the abdomen, all Opisthothelae have their spinnerets loaded up near the very tip of the abdomen, where the spigots and their silk can be easily reached by the rear-facing legs. Finally, while some Opisthothelae are nocturnal, many of them are diurnal. There's a lot more variety in the behavior and life strategies of the Opisthothelae than there is in the Mesothelae, although that's probably because there's barely more than 100 species of Mesothelae and close to 50,000 species of Opisthothelae. So, with that taken into consideration, it kinda makes sense why the Opisthothelae are so much more diverse. Now, this may seem like a pretty long list of traits that have been drastically altered between the Mesothelae and the Opisthothelae. That's true, it is a long list of traits, but it's not like it came out of nowhere, or it's not like it happened overnight. The first Mesothelae fossils are around 300 to 310 million years old, and the lineage itself is a bit older, perhaps somewhere between 310 and 330 million years old. In contrast, the oldest known Opisthothelae fossils are from the early Triassic, around 250 million years ago. Now, it's a pretty fair bet that the actual lineage is a little bit older than the oldest fossils we've found, but even still, there's a pretty large evolutionary gulf between the Mesothelae and the Opisthothelae, filled with a gradient of extinct species, of intermediaries that formed a living bridge through time, connecting one group to the next. Among the Opisthothelae, their early evolution produced two major subgroups. There's the more basal Mygalomorphae, which still retain a handful of features seen in their even older Mesothelae cousins, and then there's the more derived and much more diverse Araneomorphae. The older lineage, the Mygalomorphae, are a robust and sturdy breed of spiders. They're generally large, with thick limbs, bulbous body segments, and usually, but not always, a thick coat of bristly hair or setae that provides immense tactile sensory feedback. Famous members of the clade that I'm sure you've heard of include funnel web spiders and the tarantulas. One of the Mygalomorphs' more primitive features, which they share with their older Mesothelae cousins, are downward-pointing, vertically oriented chelicera. These were perhaps retained because the downward-pointing chelicera allowed for the application of maximum force onto a prey animal. Considering the relatively immense bulk and strength of the Mygalomorphs, a vertical stabbing or piercing motion would be strong enough to penetrate the shell, scales, or carapace of virtually any prey animal. In contrast, the Araneomorphae have diagonally oriented chelicera that move horizontally, coming together kind of like a pair of scissors. Another primitive characteristic that the Mygalomorphae share with the Mesothelae is that their respiratory structures are limited to just 2 pairs of book lungs. It might seem like 4 individual respiratory structures would be able to provide a lot of oxygen, but actually, not really. Each individual book lung really isn't that efficient. It's actually one of the reasons why the Mesothelae have such low-activity lifestyles, where they spend most of their time sitting and waiting. They just don't have the oxygen processing capacity to produce the energy necessary for active hunting, or for always being outside, roaming around and chasing after stuff. Now, depending on the species, a Mygalomorph spider may have 4 to 6 book lungs, and these are generally larger and a bit more beefed up, providing a bit more oxygen to the spider's tissues. This has enabled slightly more active lifestyles, although most Mygalomorphae still use a primitive strategy. They still build a burrow with a trapdoor, or they line the burrow with silk like a sock-like web, and they hang out inside until they sense nearby prey. Then they rush out to seize the prey before it escapes, and drags it back inside the burrow to eat it in the quiet darkness of their home. Again, in contrast, the Araneomorphae have a slightly different respiratory design. Like the other spiders, they originally had 4 book lungs, or 2 pairs of book lungs, but they've evolved to modify that second pair. The first pair of book lungs is positioned near the front of the body, but the second pair of book lungs has been transformed by evolutionary forces and adapted into a series of small tracheal tubes that permeate the body. These tracheal tubes, derived from the book lungs, are actually more efficient than the book lungs, and they provide more oxygen to the Araneomorphs than the Mygalomorphs can produce, even with a larger number of the slightly less efficient book lungs. If we were to skim through the ranks of the Mygalomorphae, we'd see many species that retain a relatively low-energy, sit-and-wait, ambush-style hunting strategy. From primitive to derived, a wide variety of Mygalomorphs, like the Hexathelidae, the Dipluridae, the Domiothelaena, the Mygidae, the Bimeridae, the Nemesiidae, the Idiopidae, the Barrichellidae, the Tenisidae, the Nemesioidina, the Halonoproctidae, the Microstigmatidae, and many others, all produce crude sheets of webbing, which they lay out in traps and or roll into a tube shape or sock-like home, typically lining the inside of a crevice or a dug-out burrow. These tube-shaped homes usually have a gate or a trap door made of silk, where the spider can jump out and attack. Other strategies include the building of classic trapdoors, where a sheet is placed over a pit or a hollow, concealed with a light covering of forest debris like moss and lichen and bits of bark and leaf detritus. Any unwary prey animal walking by might step on this sheet of silk placed over a hollow. The prey animal's weight will cause them to push through it, and they'll fall into the spider's lair. In this instance, instead of the spider darting out at you, the spider just tricks you and makes you come to it. It should be noted that some spiders prefer to actively hunt outside of their burrow, or they may be forced to do so if food is sparse. This kind of sit-and-wait trapdoor strategy is energy efficient, but it's not very proactive or nutritionally secure. Over-relying on the lazy sit-and-wait strategy often results in lower prey variety and lower nutritional quality in the spider's diet. Naturally, if the spider doesn't get off its butt and hunt down something different, this lazy feeding strategy can lead to issues like stunted growth or malnutrition and suboptimal conditions for eggs and larvae. In other words, it pays to get out there and explore, and hunt down new kinds of prey that you otherwise may not get to experience, because that provides a more nutritionally diverse diet, and that in turn leads to better growth and development, better health outcomes, like healthier eggs and offspring. Some of the more well-known burrow-building Mygalomorphs are the Australian funnel-web spiders, or the Atracids of the Atracidae family. These fearsome creatures are quite typical in the sense that they'll dig a burrow into the soil under a rock, or into the bark of a tree, or into the softening woody flesh of a decaying log. From within this burrow, they'll roll out several trip lines, but they don't usually build a trap door or a door entrance to their burrow. Their use of webbing is limited to the trip lines and the sheets that line the inside of the burrow to make it soft and cozy. So far, this is all well and good. It's all normal for a Mygalomorph. Now there's two reasons why the Australian funnel-web spider is well known. The first reason is because they possess a venom that is particularly dangerous. The well-developed chelicera contain venom glands and venom sacs that deliver a stinging slurry of atrachotoxins. Among them, delta-atrachotoxin, a potent modulator of the neuromuscular junction. They cause uncontrolled, often continuous release of acetylcholine, which activates the muscles. Normally, the nervous system releases small amounts of acetylcholine at the neuromuscular junction to activate the muscles as desired to perform coordinated movements. The spider venom causes an acetylcholine dump, and the muscles spasm, twitch, and seize, leading to general immobility and difficulty breathing, in addition to a laundry list of other awful symptoms, including high blood pressure, edema, acidosis, and death. The second reason the Australian funnel-web spider is so well known is because its well-developed chelicera are freakishly powerful. They are huge and heavily muscled, and very, very sharp. Australians report that the bite of a funnel-web spider can puncture leather shoes to stab at the feet inside. These monstrous chelicera can even pierce through fingernails or toenails to inject venom into the nail bed. Smaller animals that the funnel-web spider would actually consider viable prey include insects with flimsy chitin exoskeletons, and small reptiles and rodents with soft, thin hides. When it comes to resisting a puncturing bite from a funnel-web spider, these creatures don't stand a chance. There's another family, closely related to the Atracidae, called the Actinopodidae. These spiders are also relatively dangerous due to their size and the potency of their venom. Within the Actinopodidae, there's a striking genus called the Mesothelae, which is primarily found in Australia, although one member species is found in Chile in South America. This genus has amazing coloration, like in the Mesothelae ocatoria and the Mesothelae insignis, both of which have a vibrant red color to their head and chelicera, and a shiny gunmetal blue color to their abdomen. The closely related Actinopus genus is an older, more diverse lineage, with member species found all over South America: Peru, Argentina, Paraguay, Uruguay, Brazil, even as far north as Guyana, Colombia, and Panama. Now this gets super fascinating if you look at a map of Gondwana. This was a supercontinent that formed about 550 million years ago, lingering in the southern reaches of the southern hemisphere before breaking apart a few 100 million years later. To visualize Gondwana, imagine South America and Africa stuck back together. The landmass of Antarctica is fused with the southern edge of Africa, from Cape Horn to Madagascar. The entire Indian subcontinent is wedged in here too, between Antarctica and Somalia. Australia was fused to Antarctica, directly opposite of Africa. This massive supercontinent dominated the southern hemisphere for hundreds of millions of years before breaking up into the conformation we know today. Populations of all animal species, not just spiders, emerge on this landmass, diversified and radiated, and eventually they would find their descendants scattered among the distinct land masses, separated by vast oceans and diversifying on their own isolated worlds. This is what appears to have happened to the Actinopodidae family. The basal lineage, or the common ancestor, appears to have produced the Actinopus genus hundreds of millions of years ago when the Gondwana supercontinent was still around. The Actinopus diversified in the forested area that is now South America, with some groups reaching the mountains lining the western coast. Over hundreds of thousands and even millions of years, these diversifying groups spilled along the coastline of Gondwana, across the mountainous edges of what is now Antarctica, and onto what is now Australia. One of these groups was the Mesothelae, which originated in South America but had descendants spread across Gondwana to colonize Australia and diversify there on their own. Perhaps the largest and most famous division of Mygalomorphae is the family Theraphosidae, which includes over 1,000 species of tarantula. Although some species are rather small, the tarantulas are famous for their mini giants, including the Brazilian salmon bird-eater Lasiodora parahybana with an 11-inch leg span and an upper mass of 100 grams, and the Goliath bird-eater Theraphosa blondi with a 12-inch leg span and an upper mass of 170 grams. Despite their size and their fierce names, the tarantulas are fluid feeders, just like all the other spiders, so even though they can attack and kill relatively large prey animals like small rodents, lizards, snakes, bats, birds, and virtually any kind of terrestrial invertebrates, they still have to grind up their prey and dissolve them down into a gory soup. To grind up the bones and shred the hides of small vertebrate prey animals, the tarantula have evolved enormous, heavily muscled chelicera and robust pedipalps armed at the tips with sharp cheese grater-like plates. Interestingly, despite the presence of these very sharp food-grinding plates, the pedipalps are still used to hold sperm and insert it into the female's genital pore during sexual reproduction. Although, from the research that I've been looking at, it doesn't really look like the presence of these food-slicing plates on the pedipalps causes any appreciable damage to the female during reproduction. With regards to all of their other qualities, compared to other spiders, tarantulas generally have poor vision. They can't really form images with their eyes, and they can only detect coarse changes in lighting and movement. Their perception of the world is reliant on sensing vibrations in the air or the soil, and the smells emitted by prey animals and predators, and the pheromones of potential mates. Like the more modern Araneomorphae, the tarantulas have spinnerets at the far tip of their opisthosoma, but the tarantulas have just 2 to 4 spinnerets, where the Araneomorphae usually have 6. Tarantulas also just have book lungs, and none of the fancier, more derived tracheal networks seen in the Araneomorphae. A really fascinating defensive trait seen in tarantulas is a kind of rearing behavior. When they feel threatened, they'll lift up their cephalothorax and their front legs, they'll spread their chelicera wide, and if possible, they'll make a loud, stridulating hissing noise to scare off the threat. If the predator is not discouraged and continues to approach and get closer, the tarantula may slam down its front legs against the predator to try to startle it and scare it off, kind of like giving somebody a sucker punch or a sudden slap to the cheek. If this doesn't scare off the predator, which continues to approach and engage the tarantula, then the tarantula can respond in one of several ways. It may just try to run away. It might conclude, okay, my initial methods to discourage the predator haven't worked, it's still coming at me, I don't have time to do anything else, so let's just get out of here, and it'll run away, and most likely hide in a crevice or a hole somewhere until the predator gets bored and decides to go do something else. Alternatively, the tarantula can attempt a dry bite. A dry bite is when the tarantula lashes out with its chelicera and tries to bite the predator or the target, but does not inject it with venom. Venom is very expensive. It's very metabolically expensive to produce. So if you can scare off a threat with a dry bite without having to spend some of your precious venom, that's really valuable. That can save a lot of time and energy. And also, if you get attacked by another predator in short succession, if you scared off the first one with a dry bite, then that means you still have venom that you can use for this new predator. But on the other hand, if the spider does use its venom, if it does use a wet bite to scare off the predator, say it works, say it does scare off the predator and it's been injected with venom and it's suffering the pain and the sting and the muscle paralysis and all the stuff that comes with that, well, what if a second predator comes by? Well, the tarantula just depleted all of its venom. It's got none left. So if it's going to make any kind of attack on this second predator, it's going to have to be a dry bite, and it's going to have to hope that that's enough to scare off the predator. At least until, of course, it can regenerate its venom stores. A tarantula will generally only use its venom on a predator if it's really desperate. If it feels like it's its last resort, it's its only option, otherwise the predator is definitely going to kill it. The tarantula would much rather use its venom for capturing food. Perhaps the trait most unique to the tarantulas are their urticating hairs. In addition to the setae, the bristles all over the body used to detect vibrations, the tarantulas also have these other kinds of specialized bristles growing out of their abdomen. The urticating hairs are a defensive feature. They're designed to be rubbed, scraped, or abraded off the abdomen and kicked towards the predator. The urticating hairs are very fine but very stiff bristles, equipped with brutal hooks and barbs. The purpose of these urticating hairs, which are basically like nails dumped onto the freeway, is to get into the eyes and mucous membranes of a predator and cause intense irritation and discomfort. They're legitimately dangerous objects, as small predators like mice can actually be killed by urticating hairs, for example, by clogging their nose or getting into their lungs. The tarantula, in turn, must use these urticating hairs carefully, because they don't grow back. They get replaced during a molt, but molting doesn't happen too often, so the urticating hairs have to be used sparingly. They have to be used very strategically. Some species of tarantula, like those in the New World, don't have urticating hairs. To compensate for the lack of this defensive weapon, the New World tarantulas are generally more aggressive, and they produce larger quantities of more dangerous venom. In other words, because they have a lower number of defensive and offensive weapons, they take the weapons they do have, like their aggressive behavior and their venom, and they really soup it up. They upgrade it to make it more powerful or more extreme. Alright, so we just looked at several 100 million years of arachnid evolution, from the ancestral Planeturga 470 million years ago, to the true arachnids, which emerged about 435 million years ago, to the first spiders, the first Mesothelae fossils around 300 to 310 million years ago, to the first Opisthothelae, which emerged around 250 million years ago. The Opisthothelae produced two major groups, the Mygalomorphae, like the funnel web spiders and the Tarantulas, which we've explored in great detail in this episode, and the Araneomorphae, which up until now I've only mentioned a few times in passing or to make comparisons here and there. The Araneomorphae includes the vast majority of spider species, and we'll be exploring them in the next episode, in part 2 of this mini-series on the spiders. As we went over a lot of spider anatomy and physiology and evolution in this episode, for the next episode, for part 2, we'll focus pretty much entirely on the spiders themselves. We'll explore their biodiversity, their ecology, their predator-prey dynamics, all of that fun stuff. And if that sounds cool to you, then hit the subscribe button. If you enjoyed this episode, then hit the like button. Or if you're listening through a podcast app, then give the show a 5-star review. I hope you can join me in the next episode, part 2, in this spider mini-series, as I explore the Araneomorphae. And as always, thank you for listening.